what I've been interested in for many years now, is how does the precise positioning of the flower at the shoot tip, there's a structure that we call the meristem, where there are stem cells, okay, that have the ability to generate new cells all the time, and this gives the property to this tissue to make the flowers, okay? But the tissue doesn't make the flower at random position, okay? It positions them very precisely, accordingly to the other one that are existing. And so then the problem is very similar to what I was describing before, you know, this problem of emergence, is that we think that the way this works, it gets positioned at a certain place because there are signals exchanged between all the flowers that exist, okay, and the rest that will canalize the system, into positioning the flower at this position, okay, and actually also with a certain frequency. The type of phyllotaxis that Arabidopsis has is a spiral phyllotaxis, meaning that the flowers are produced one by one, okay, and then distributed along the stem so that they produce a spiral like that. A spiral can be perfect, meaning that it really follow perfectly the spiral. But you can have also errors that could be small deviations, okay, from what you expect, because the angle between successive flowers is quite well controlled. Huh? So if you measure most plants like this that have a spiral phyllotaxis, the angle in between flowers will be close to 137. Okay, so you can have small deviation of this angle, but you can have also, if you want, mistakes in the sequence of angles. And so when you have this kind of observation, one thing you could imagine doing is like it's taking this, using this to actually go back to the genetic origin of why you don't have the same robustness. And so then the big biological question, very fundamental, is really, can I find genes that control the robustness of the architecture of an organism? And in the frame of ROMI, this is really typically something we can do if we have a phenotyping station and pipeline for analysis. That actually works well, because, and if it's high throughput, because then what you can do, if you want to make it simple, you can take these plants from two different origins, you cross them, and then in the progeny you can analyze them so that to know what is how is the phyllotaxis, how robust it is, okay? And so it's actually not the plants themselves, you go for their progeny and then you look in the populations, and then you can go and map the origin of the genetic origin of the control. But for this you need to look at a lot of plants.
So here basically we will enter the culture chambers, and so in those culture chambers we are culturing all kinds of plants, so Arabidopsis thaliana. As I told you, I'm interested in phyllotaxis, which is the angle between two successive organs. And this is not random, and the plants build each new organs with a very precise fashion, with a lot of regularities. But sometimes they make some errors, mistake, which are very indicative of how the plants built this. And also you can have mutants. Then you invalidate the function of one gene, and then you change the phenotype again. And then by this change in phenotype, you can try to go back to the function of the gene. But for this, you need to measure the angles. So to precisely measure it, so you need to not to measure it only on two organs. You need a lot of plants, a lot of organs. So it's a lot of tedious measurements. So what I'm doing now is I'm doing it by hand. So I take one plant, I put it here, down here at the top, just to maintain it. I'm stretching it a bit, and here I have a protractor, and with this protractor I move it along from organs to organs, and I just measure the angles between successive organs. So this is really built for Arabidopsis thaliana, for example. But if I would have another plant, for example, this would not be appropriated. So you can see that it's very tedious. Like for one plant, it takes me like more than 20 minutes. And then if I want to have like the length between internodes, I need to go to another tools to measure it. So it's never ending. But that's how we did now. So we would really be happy to have like a tool which is able to to grasp the 3D architecture in all directions, so that we have an object where we can turn around virtually, which comes from the true plant, and then we can make accurate measurements of the angles, of the lengths. And then I'm not like a specialist in one plant, because if I have another plant, like a Chenopodium, that would not fit in this device because this is too tiny, it's only built for Arabidopsis. But then with my robot, with the robot 3D, we can turn around and it can accept a lot of plants with a lot of different architecture. So this is like the prehistoric tool, and with ROMI we hope to move to the to the 20th century at last. What could be different here? So nobody walking here, but the researcher in his natural environment.
Exactly.
Like and, so for example, you can see here like it's 6 plants, and they have been growing in the same environment, so very close together, and yet you have plants that are bigger and plants that are smaller, for example. How do you explain this? Is it because a gene is different, or for a farmer, is it because this slot here is too humid or not enough, or but at least there is something different. The phenotype is different. The information that you get from what your plants look like is very important. And again, like you can do it with your eye, but if I have to do it for all the plants here, like a farmer would have to do for all the fields, it will take a lot of time. So if you can have a robot that say, oh, like a quantify measure, like 80% of your plants have retardation. 80%, it's not 5%. So then, okay, I have to postpone my harvest, for example.
So Fabrice, the biologist of the project, is interested in organ arrangement around the stem. So as you can see here, we have the main stem, and we have lateral organ arranged around the stem. But as you understand, if you try and measure this by hand, this is first not reproducible, and second, subject to the experimenter capacity to really look into proper detail or to take up as much information as possible. Whereas if we do numerical reconstruction of the plant, once you've done it, you can apply a succession of different algorithms. One would be to compute the angle between the successive organ, but if like 5 years later you're interested in the internode length, you can take back this 3D reconstruction you've made and then apply another algorithm that will quantify this internode length. So by transforming the biological data into numerical data, you can keep on working with the same data almost forever.
That's
quite cool, right?
That's really cool.
That's really good.
This kind of knowledge that we're generating, I mean, is really fundamental. Huh? It's I think the kind of question we're addressing is really, I mean, almost at the essence of life. Is how do you construct an organism? And this is it's true for plants, it's true for humans, for all animals. Is that you start from one cell? Okay, the plants and animals are the same. I mean. And then you're going to construct very complex structure from that. So how can we go from something so simple to something so complex? Okay, so it's, I mean, yeah, it's a dream to understand this, because you understand this, you understand how, at least part of how life is constructed. I mean, and maybe how it evolved on Earth, how you diversify the shapes, etc., etc.